Dear audience, I would like to speak about a transposon-mediated gene therapy system in a mouse model of tyrosinemia type 1. Tyrosinemia type 1 is a fatal genetic disease. It is caused by the germline point mutations of the fumaryl acetoacetate hydrolase gene. The product of this gene is the last enzyme in the tyrosine degradation pathway. So the lack of this enzyme causes the accumulation of fumaryl acetoacetate and mallyl acetoacetate, which are converted to a highly hepatotoxic metabolite called succinylacetone. This condition can lead to severe liver damage. There is a pharmacological inhibitor called NTBC, which is able to block the tyrosine degradation pathway upstream of FAH because it blocks the enzyme called HBD, and it prevents the accumulation of the toxic metabolites, so it can prevent from the acute liver injury. We have created the mouse model of the disease with the deletion of the significant part of the FAH gene in a mouse stain. We use this stain to perform different gene therapeutic preclinical model experiments. In our experiments, we use the high regeneration capacity of the liver because 10% intact tissue is able to regenerate the complete organ. So we isolate hepatocytes from mouse suffering from the tyrosinemia type 1, and we perform different genetic corrections in the cells, and after that we inject the corrected cells into another diseased animal, which is an NTBC treatment from the beginning of its life. So when we, after the transplantation, we withdraw the NTBC from the animal, so the transplanted cells gain a growth advantage, and they are able to repopulate the liver. So we gain a transgenic organ, which is able to tyrosine degradation. Through our experiments, we use multi-step collagenase perfusion, where we use the animal's own cardiovascular system in order to isolate the hepatocytes from the intact liver from the animal. After the perfusion, we use Perkol gradient centrifugation, where we can isolate just the hepatocyte fraction, and after that we make the genetic correction on the hepatocytes. After that, we inject these corrected cells into the spleen of the animal, and from the spleen through the lienal vein and then through the portal vein, the cells migrate to the liver, where they can proliferate and repopulate it. In our ex vivo system, we electroporate the isolated hepatocytes with two plasmid constructions. The first construction contains the Sleeping Beauty transposon inverted terminal repeats, and between the two ITRs there is an M-cherry fluorescent marker gene and a therapeutic FAH gene linked with a T2A peptide gene, which enables the ribosome skipping through the translation of these two separate proteins. The second plasmid expressing a hyperactive version of the Sleeping Beauty transposase. The transposase catalyzes the transposition reaction, which is a simple cut and paste mechanism where this sequence between the inverted terminal repeats is cut from the plasmid, and then the transposase enzyme integrate this sequence into the genome of the animal. It only requires a TA dinucleotide in the genome, which duplicates through the transposition reaction. These potentially corrected cells are injected to the spleen of the animal, and after the removal of the NTBC, these cells gain the growth advantage, they co-express the fluorescent marker gene and the therapeutic gene. The weight curve shows that the placebo-treated animals go through a progressive weight loss, and they die within 5 weeks. But the treated animals are, after mild weight loss, progressively gaining weight, and they reach the normal average weight of the wild-type control animals. And it is visible that after 2 months, almost the whole liver show that fluorescent signals. So the efficiency of the gene therapy is really high. We took samples from these regenerated liver tissue and isolated genomic DNA from it in order to identify the transposition sites. So we used the PCR-based method. It's called transposon display in order to analyze the transposition reactions. In the transposon display, first we digested the genomic DNA with restriction and the nuclease DPN2. It cuts the genome frequently, and then we used that fragments which are containing the end of the therapeutic construction with the Sleeping Beauty inverted terminal repeats and the flanking genomic regions. So we ligated DPN2 adapter oligonucleotides to the end of these fragments, and we performed touchdown PCR where we used biotonated primers and amplificated this sequence. After the first reaction, we cleared the amplificated sequences with streptavidin pairs, and after that we performed the second PCR where we amplified a tighter region in order to amplify just the sequences with real transposition sites. After that, we sent our amplified samples to high throughput sequencing. Then we analyzed the data, and we were able to identify each integration site. And after that, we could state that the integration pattern of the Sleeping Beauty was really close to random, because we set the random baseline where the presence of the TA dinucleotides in the genome. So 45% of the TAs were in some annotated genomic regions, and compared to this, 63% of our Sleeping Beauty integrations were in annotated genomic regions. It is much more better than any retrovirus, lentivirus vectors, which are frequently used in different gene therapeutic approach. So, but the efficiency is retrovirus-like, and the risk for the insertional oncogenesis is substantially smaller. So maybe our Sleeping Beauty transposon system is able to be a promising gene therapeutic approach in the future. I would like to acknowledge my mentor and my supervisor and all the staff members in the Tumor Genome Research Group for their generous help. And I would like to thank you for your attention.